Bismillahirrahmanirrahim. This video is about the morphological features of sarcomere and its role in the skeletal muscle contraction. Let's begin. Skeletal muscle cells are long and cylindrical, so they are commonly referred to as muscle fibers. The skeletal muscle fibers have striated appearance due to the arrangement of the contractile myofilaments of actin and myosin in a sequential order. The sarcomere is the functional unit of the muscle fiber. A sarcomere is defined as the segment between two neighboring parallel Z lines. The Z lines are anchoring sites for actin and myosin filaments. Surrounding the Z line is the I band, the region where actin filaments are not superimposed by myosin filaments. The region between two neighboring parallel I bands is known as the A band and contains the entire length of single myosin filaments superimposed by actin. Within the A band is a region known as the H band and is the region not superimposed by actin filaments. Within the H band is the M line, which is composed of myosin, myofilaments and titin molecules. The contractile myofilaments within the muscle fiber are arranged in repeated thick and thin filaments. Myosin is the main component of the thick filaments and functions as a motor protein. Each myosin molecule is shaped like two golf clubs twisted together. The myosin tail is shaped like twisted golf club handles. Tails of neighboring myosin molecules lie parallel to one another, forming the shaft of the thick filament. The two projections of each myosin molecule are called myosin heads. Each myosin head has two binding sites, one for binding actin and second for binding ATP. The ATP binding site also functions as an ATPase enzyme that hydrolyzes ATP to generate energy for muscle contraction. The main component of the thin filament is the protein called actin. Individual actin molecules join to form an actin filament that is twisted into a helix. On each actin molecule is a myosin binding site where a myosin head can attach. Smaller amounts of two regulatory proteins, tropomyosin and troponin, are also part of the thin filament. In relaxed muscle, myosin is blocked from binding to actin because strands of tropomyosin cover the myosin binding sites on actin. The tropomyosin strands, in turn, are held in place by troponin molecules. To fully understand the skeletal muscle contraction, you must watch my video on excitation-contraction coupling at the neuromuscular junction. The link of that video is available in the description. If you have already watched that video, let's go ahead with this one. The sequence of events that result in the contraction of an individual muscle fiber begins with the release of acetylcholine from the motor neuron innervating that fiber. The local membrane of the fiber will depolarize as positively charged sodium ions enter, triggering an action potential that spreads to the rest of the membrane, including the T-tubules. This triggers the release of calcium ions from storage in the sarcoplasmic reticulum. The calcium ions then initiates contraction, which is sustained by ATP. As long as calcium ions remain in the sarcoplasm to bind to troponin, it keeps the actin binding sites uncovered to interact with myosin. Additionally, as long as ATP is available to drive the pulling of actin strands by myosin, the muscle fiber will continue to shorten to an anatomical limit. Muscle contraction usually stops when action potential from the motor neuron ends, resulting in repolarization of the sarcolemma and T-tubules and closure of the voltage-gated calcium channels in the sarcoplasmic reticulum. Calcium ions are then pumped back into the sarcoplasmic reticulum, which causes the tropomyosin to again cover the binding sites on the actin strands. A muscle also can stop contracting when it runs out of ATP and becomes fatigued. Thanks for watching and listening. Do share this video with your friends and colleagues. If you like this video, subscribe to this channel for watching more videos.